For this topic, we're going to be talking about enzymes. And it's good to start with thinking about what enzymes are. So enzymes are biological catalysts. And what this means is that as catalysts, they will speed up the rate of a reaction without themselves actually being used up or chemically changed. And these can be all different kinds of reactions. We will look at a few different examples of reactions, but it's good to kind of have in your mind that pretty much all reactions of any kind that occur in living organisms are catalyzed by enzymes. So they need to be sped up so that they can actually take place at a rate that's actually fast enough. So we describe them as biological catalysts because they are catalysts found in living organisms. We think about what they actually are. So enzymes are globular proteins. So that links to the idea of proteins being either mostly globular or fibrous. As globular proteins, they will then have often hydrophilic R groups on the outside, but they'll generally have a very complex tertiary structure. So thinking about our levels of protein structure, so tertiary structure, they can have a quaternary structure as well, and lots of enzymes will, and that will be defined by whether or not they've got more than one polypeptide chain as part of the structure of the enzyme. Enzymes are often involved in quite complex metabolic pathways. So what that means is that it's a cascade of reactions, or a cascade of enzyme-controlled reactions, where there's a kind of a start point and an end point, and there are lots of steps along the way that are all of those individual steps are going to be catalyzed by individual and specific enzymes. There's two kind of main groups of where we might talk about where enzymes actually work, which is that they are either intracellular or extracellular. Catalase is our example of an intracellular enzyme. So intracellular enzymes are produced and function within a cell. So they're both synthesized, they're both produced as proteins inside of the cell, and then their function is also carried out inside of the cell. Catalase, which is used really frequently for practicals, catalyzes the breakdown of hydrogen peroxide into water and oxygen. That's it as a formula balanced. This is useful because hydrogen peroxide is quite a common waste product of a lot of metabolic reactions. So a lot of cellular reactions will produce hydrogen peroxide as a waste product of that. Amylase then is an example of an extracellular enzyme. Extracellular enzymes are secreted by cells to catalyze a reaction that is outside of the cell, so to catalyze an extracellular reaction. Amylase specifically hydrolyzes starch, so amylase catalyzes the breakdown of starch into simple sugars. We've really talked about the role of enzymes kind of together with that one, so we'll move past that and go straight to talking about the mechanism of enzyme actions. When we're talking about enzymes, it's important to remember that they are proteins with a very specific tertiary structure. When we get on to talking about the effects of pH and temperature, is this idea of linking to the effect that they have on the tertiary structure and a very specific three-dimensional shape. And that three-dimensional shape is held together by the different types of bonding that we see at the tertiary structural level. So hydrogen, ionic, disulfide bridges, and hydrophilic interactions. A really, really important key term when we're talking about enzymes is talking about the active site, because this is the part of the enzyme where the substrate will actually bind to. When the substrate binds to the active site of an enzyme is that we use the term enzyme substrate complex. So if you're looking to describe when the substrate has actually successfully bound to the enzyme, is you could talk about the formation of enzyme substrate complexes, whether that's possible or not, and that's going to be referred to when the substrate is bound to the active site. Active sites themselves are very, very specific in their shape so that they're able to fit the substrate. So one enzyme will likely only work to catalyze one really specific reaction with one very specific substrate, and even a small change to the chemical structure of that substrate could mean that then the enzyme's no longer effective at all. There are two different hypotheses that we use to try to describe the mechanism of enzyme action. So the first one is the lock and key hypothesis. This will be one that people are likely familiar with from GCSE or kind of prior education before sixth form. And in this hypothesis, the enzymes have this highly specific three-dimensional shape that is going to fit the substrate really precisely and really rigidly, in the same way that a metal lock and a metal key, they fit together and they fit kind of perfectly and precisely, but they are very rigid because pieces of metal are very rigid. There are some parts of this that we kind of do carry forward to the second hypothesis, the idea that they are highly specific and have a three-dimensional shape. Definitely that is common to the second hypothesis, but the idea of being rigid is not. So the second hypothesis is the induced fit hypothesis. In this hypothesis, the enzyme and the substrate do have highly specific three-dimensional shapes, but that shape can change slightly when the substrate binds to the enzyme. So the presence of the substrate in the active site can cause some small changes in shape of the enzyme as it binds. This idea of the enzyme, or really of any molecule, changing shape slightly as it binds is a conformational change. So it's not a kind of chemical change to it, it's just a slight readjusting of the structure of it as the substrate enters the active site. So the enzyme will change shape slightly, a little bit more like kind of hand and glove or foot and shoe rather than lock and key. It also involves a bit of temporary binding as the substrate is in the active site of the enzyme. So that's part of it being an enzyme substrate complex with this hypothesis. So the next term on there to look at is the idea of an enzyme product complex. So whilst the substrate's bound to the enzyme and then it is converted into product, there'll be a kind of brief time where the product is still bound to the enzyme, still in the active site. So that's enzyme product complex. And then when that product gets released is then that's product formation. So the last term there on here to talk about is the idea of activation energy and of enzymes' ability and their action, their mechanism, being to do with lowering this activation energy. So we kind of need to start with having an understanding of what activation energy actually is. So all reactions require some energy to take place. So the activation energy of a reaction is how much energy is required for the reaction to take place or to initiate, because some reactions will take in energy kind of overall the whole reaction. Some of them might give out energy as part of the reaction. If something is exothermic, then it might give out energy across the whole reaction, but it will still need some activation energy to actually start the reaction. And this might be kind of kinetic energy through kind of stirring or mixing. It might be an increase in temperature instead. What enzymes are doing, though, is that the enzymes are going to destabilize the bonds within the substrate. So as the substrate is in the active site of the enzyme is destabilizing some of those bonds or kind of holding the substrate in the correct position for the reaction to be able to take place more easily. So it's providing an alternative pathway. So that's a term that we use when we talk about enzymes lowering the activation energy for a reaction, is that they're providing an alternative pathway. What this might look like in practice, though, is that a reaction could take place maybe at a lower temperature, such as body temperature. So lots and lots of reactions in living organisms would require very high temperatures to take place outside of a living organism and outside of the presence of enzymes. So enzymes make it possible for those reactions to happen at temperatures that's possible for life. Lots of different factors affect the action of enzymes and the rate of enzyme reactions. And when we're talking about pH and temperature, then we talk about them having optimums. So all enzymes have an optimum pH. Extreme pHs, now the idea of it being extreme is important. So we're talking about pH changes that are not just a little bit away from the optimum, but maybe a couple of kind of pH away from the optimum can denature enzymes. A higher pH, or what we might describe as being alkali, or a basic pH, what that actually means is that there are more OH minus or hydroxide ions present, whereas a low pH, or what we would say as acidic pHs, have got more H plus or more hydrogen ions present, and those charged ions can interfere with and can break hydrogen bonding and ionic bonding. So the kind of weaker bonds that exist within the protein and are forming part of the tertiary structure bonding is that those can be broken. So what's happening is that really specific 3D shape that we talked about already can be changed, and specifically it can change the shape of the active site. So really good to be specifically mentioning the active site here. And what does that actually mean? Well, what that means is that enzyme substrate complexes can no longer form. So the substrate doesn't fit into the active site anymore. Their shape is no longer complementary. Using either of the hypotheses, either lock and key or induced fit, is they no longer fit. This is a good time as well to mention solutions that are called buffers. So they come up a little bit more often in chemistry than in biology, but a buffer is any solution that is able to maintain a specific pH. Now, they don't work indefinitely. If a reaction is taking place that's releasing hydrogen ions over a really kind of long time frame, it's producing loads of hydrogen ions, eventually the buffer won't work anymore, but they're able to hold the pH at that kind of specific point for a reasonable duration of the reaction. Moving on to temperature then. So all enzymes will also have an optimum temperature. With temperature, it's slightly more complicated than just saying extreme, because actually what's going on is slightly different at lower temperature to higher temperatures. At lower temperatures, there is a lot less kinetic energy. So the enzyme, the substrate in solution, are moving around much, much more slowly, and so they are going to be colliding much less often. So a really useful phrase for describing this is to talk about there being less frequent successful collisions. So a successful collision being when the enzyme and the substrate collide together in the correct orientation, where the substrate can actually go into the active site. You can also talk about them colliding with less energy, and maybe therefore it not being enough energy to meet that even lowered activation energy. As you start to raise the temperature from it being too low, the rate of reaction will be faster. However, once we start to reach much higher temperatures, and temperatures that are more extreme and that exceed the optimum temperature, is that we would start to see denaturation occurring. So the enzymes would start to denature. Their kinetic energy, that excess kinetic energy, what it will do is, rather than just making them keep kind of colliding more and more frequently, is that it causes the bonds to vibrate and to have kind of strain put on them. And that will, similar to with pH, particularly affect the weaker bonding that we see as parts of the tertiary structure of the enzyme, which means that it's going to primarily affect hydrogen bonding and ionic bonding. Similarly to when we're talking about extreme pHs, so the shape of the active site is permanently damaged. A way to visualize this is, if you're kind of frying an egg, is that once the temperature is extreme, it far exceeds the optimum, is that the shape of the active site and the structure is permanently damaged. So the substrate cannot bind. It's not reversible. You can't lower the temperature and it uncook the egg. You cannot undenature a protein just by lowering the temperature of it. The next bit on here to talk about is Q10, or the temperature coefficient of an enzyme reaction. If the temperature coefficient, or Q10, is 2, what that means is that the rate of reaction of an enzyme catalyzed reaction is doubling every 10 degrees. Now, this is always talking about temperatures up to the optimum, because of course, once the temperature goes very high and extremely high, then we would see denaturation. The formula for this is simpler than it looks when it's written down. So to work out the Q10 is that you would read off of a graph the rate at a temperature that is 10 degrees higher, or x plus 10 degrees, and you would divide that by the temperature that's 10 degrees lower. For an example of this is that you could maybe look at the calculating the temperature coefficient using the rate at 30 degrees, so that's your rate at x plus 10, and at 20 degrees, so that would be your rate at x degrees C. I've added some graphs in now for this one, where you can see for the temperature graph that we've got that increase from where it is low temperature up to the optimum, and then denaturation at the extreme temperatures. You can see this asymmetry, so it's not a symmetrical graph there. This graph again shows the calculation of temperature coefficient, or Q10. So we can see there that 20 degrees here again is used as an example. It doesn't have to be 20 and 30. It just needs to be two temperatures that are both lower than the optimum, that are 10 degrees apart for this. So if you're working out for this graph specifically, it would be your rate at the higher temperature, which is 20, divided by your rate at the lower temperature, which is 10, which gives you a temperature coefficient of 2. Most temperature coefficients for enzymes will be close to that. So there's two other factors which will affect the rate of enzyme reaction. So enzyme concentration, so how many enzymes are actually present. So long as there is always sufficient substrate, so long as there's always an excess of substrate, is adding more enzymes will increase the rate of reaction. So long as there is plenty of substrate available, is that adding more enzymes to that solution will mean that more enzyme substrate complexes form for that. So we would expect it to be linear until you start to run out of substrate. Substrate concentration, we always see this graph where it plateaus for this. So as substrate concentration increases, the rate of the reaction does increase, but up to a point, which you can see there is marked by the cross, and then after it is highlighted. Because once you get beyond this point, so all the active sites are in constant usage. So in this situation, the concentration of enzymes is going to stay the same. So it's constant throughout this, and more substrate is being added. At the start, definitely though, if you add more substrate, then there will be more enzyme substrate complexes able to form. So more of the enzymes will be able to be used at any one point. We will talk about practicals at a later stage for this one, because there's not a specific practical that's kind of essential for this, although catalase is really common. The last two sections for this topic is talking about coenzymes, cofactors, and then inhibitors. So first to talk about coenzymes and cofactors. So they are always non-protein. So they are not made of protein. They will be much smaller and they'll be made of non-protein parts. So they're not made of amino acids. A cofactor or coenzyme is something that is required, though, for the enzyme to function properly. So whether that means that the enzyme wouldn't work at all without it, or if it would just be very slow. The example of this picture here that I'll be kind of writing around, that is not going to work at all without the presence of that cofactor or coenzyme, because that substrate just absolutely does not fit into the active site without it. So cofactors, first of all, tend to be inorganic ions, so they tend to be very small. The big example here is chloride ions, which are a cofactor for amylase. There's no real set rule of how they have to work to be defined as a cofactor. It could be that the cofactor is required for binding at the active sites. It could be that it actually slots into the active site along with the substrate in order for the substrate to be able to bind. But it could also be that actually the cofactor binds somewhere else to the enzyme, and when it binds, it means that the tertiary structure of the enzyme changes slightly, and it's only then that it's actually complementary to the substrate. So it doesn't have to bind to the active site in order for it to affect the binding. It could bind somewhere else. And as I said, amylase is the example here. So amylase as an enzyme cannot break down starch very effectively at all without the presence of chloride ions. Coenzymes, they are a type of cofactor. So coenzymes, when we use that term, they are usually larger. So we're usually talking about larger molecules than just a single inorganic ion. And they are generally derived from vitamins. So vitamin B, for example, is where a lot of coenzymes are derived from in humans. Again, there's no set rule about exactly how they have to work to be defined as a coenzyme. It could be that actually they bind permanently to the enzyme, so then they'd be functioning as a prosthetic group. So prosthetic group was a word we came across when we were talking about levels of protein structure. So it could be that they bind permanently, or it could be that it is temporary. Coenzymes can also be involved in carrying of electrons. So that's something that we see in respiration and photosynthesis, or it could be that they are carrying other substrates or kind of intermediates of a chain of reactions between different enzymes. We will come across lots of examples of coenzymes in upper sixth when we cover respiration and photosynthesis. So this might seem a bit kind of conceptual at this stage, but we'll see some examples of coenzymes which transport electrons and hydrogen ions and also some other kind of substrates as well. So we'll see some different examples there. The last part here then is to talk about inhibitors. So there's two different types of inhibitors. They can be competitive or non-competitive. Competitive inhibitors, they compete with the substrate for the active site. So really important term here is to describe them as having a similar shape to the substrate. They will not have the same shape as the substrate. They'll still need to be complementary to the active site, or at least part of the active site. So they're going to have at least part of it that is similar in shape. Non-competitive inhibitors bind somewhere else. They'll bind to an alternative binding site. You can use the term allosteric site to describe an alternate binding site. And when they bind, it's going to alter the shape of the active site. So you can see kind of here where it binds at the site, it kind of pulls back the enzyme and it's distorting the shape of the active site. So the end result is going to be the same, that the substrate cannot bind to the active site. So it's going to be preventing the formation of enzyme substrate complexes either way. It's important to be able to identify what type of inhibitor it is from a graph. So the purple line that's labeled as the normal should look the same as our substrate concentration graph above. Competitive inhibitors, which is the green line, the top line, because they can kind of outcompete the inhibitor as you increase substrate concentration, is eventually the rate of reaction will reach very similar to the normal enzyme reaction. Because non-competitive inhibitors, they're not competing, they bind to the alternative site, they bind somewhere else, it's not possible for the substrate to outcompete it in some way. It will always be preventing the substrate from binding, so we'll see this lowered maximum rate of reaction. The very last part of this then is the idea of end product inhibition. So if you've got a chain of enzyme catalyzed reactions, in this case it's only 3, it could be much longer than this, is that it might be that actually too much product is not optimal for that organism, or it might be that actually keeping some reserves or keeping some supplies for the substrate might be important. It could be that that substrate can be used for another reaction pathway instead. So in any case where you've got end product inhibition, is that the end product inhibits an earlier step. Now it will be almost always a non-competitive inhibitor, so it won't be binding to the active sites because it's changed quite a lot from the original substrate. So it's very unlikely you'd have an end product that would still be complementary to that same active site, and it will be reversible because otherwise this can't be kind of undone, and it will form part of a feedback loop for this. It's important for kind of slowing down product formation and making sure that any excess product isn't being made.